Hello everyone, myself Priyanka Vijay Kulkarni. This video is about the transgenic plant, okay? And you will see what are the transgenic plants, what are the examples, and what plasmids are responsible to make this transgenic plant. First of all, we'll see what is meant by the transformation. Transformation, it is a process of obtaining the transgenic plant, okay? And what is meant by the transgenic plant? Transgenic, okay? Genic means gene, trans means another, okay? So means the plant with another foreign gene, okay, from the another plant or the animal which is incorporated into their chromosome. It is known as the transgenic plant. Okay, so probably yes, there is a only one natural transgenic we can say genetic transfer method is there, agarobacterium mediated gene transfer. Okay, so this agarobacterium mediated gene transfer, this causes the particular disease to the plant, and that disease is known as the crown gall disease. Okay, and it is discovered by the Smith and Towson in 1907. And what is responsible for this disease, or what material it is transferred from this agarobacterium to the plant? So that material is nothing but the plasmid. Plasmid means what? Extra chromosomal material. Okay, so this plasmid, there are one type of the plasmid, T, okay, Ti plasmid and Ri plasmid. Ti plasmid, it is responsible to form the crown gall disease, okay, and Ri plasmid, it is responsible to form the hairy root disease in the plant. Okay, yes, so here one scientist name is mentioned, Montagu and Schell. He discovered the gene transfer mechanism between the agarobacterium and the plant. He only discovered what? Transfer mechanism. Okay, then which resulted in the development of the methods to alter the bacterium into an efficient delivery system of genetic engineering in plants. Means what? I told you, there is a only natural way to transfer the gene from one to the another. There is only one natural way, and that is nothing but the agarobacterium mediated. So basically, what is this agarobacterium? Agarobacterium is okay, from its name you can conclude that it is a bacteria, okay, but what is its nature? It is a gram negative soil borne and rod shaped bacteria which is generally found in the soil, okay, and this bacteria will infect to the probably dicot plants, okay, and some monocots which has ability to release the acetoserinone. Yes, yeah, so this property is utilized by this Montagu and Schell to discover the genetically engineered transgenic plant. Okay, so we'll see what is its mean in our future slides. So most common genes in transgenic or biotic crops, these are like herbicide resistance, then insecticide resistance, then bt cotton, okay, bt gene in the field of corn, then virus resistant genes, okay. These are some common genes or traits which are used in the transgenic plants. So there are transformation methods. There are different kinds of the transformation method. Biological means natural, okay, and physical and chemical. So in case of the physical, micro injection, okay, as you know that there is a particular micro injection, and if you have to insert particular your foreign gene into the particular plasmid or particular plant or animal or anything, then you can take that gene into this micro injection, and you can directly physically inject that gene to the your what we can say, your host. Okay, then by pressure, yes, you can transfer your gene of interest to the to your host by means of the pressure. Then biolistic gene gun. Okay, in this case what happened, or we can say it is as a particle bombardment, means whatever your foreign gene, okay, or your gene of interest, you have to take your gene of interest, then you have to coat that gene of interest to in by the particular metal like the gold, okay, or yes, there is another platinum, okay, gold or platinum. By this metal you can coat your gene of interest, and by this gene gun or particle bombardment method you can directly shoot your host. Okay, then electroporation means what? Means you have to take your host cell, okay, and uh, you have to take your host cell into particular medium, and you have to place your gene of interest into that medium only. Okay, and then you have to give a particular electrical current. So due to this electrical current, or in order to this electrical current, this foreign gene will get enter into that particular host cell. Then silica carbon fibers, okay, this is another method, laser mediated. Then by using the laser light you can do this. And then there is a sat technique. Okay, these are some physical methods for the plant transformation. Then chemical methods, PEG. Okay, means chemical method means what? In this method you have to use these chemicals for the transfer of your gene of interest to your plant cells. Okay, so PEG method. Okay, then DEAE dextran, then calcium phosphate, then artificial lipids, proteins, then dendrimers. Okay, these are some things you can use for this transfer purpose. Biological means natural method. Okay, natural means what? Biological means what? Your transferring method or your vector, it should be a living thing. Biological means what? It should be a living thing. Physical chemical means what? In case of chemical you are using the chemicals. In case of physical you are using something weapon, or you are using something material to transfer. But in case of biological you have to use something living material. And what is the living material? Bacteria. Okay, so here you can use the two types of the bacteria, Agarobacterium tumefaciens and Agarobacterium rhizogenes. And there is another method also, virus mediated method also. Okay, so I told you already what is this Agarobacterium. Agarobacterium, it is gram negative soil borne bacteria which is in a rod shape. Okay, so this Agarobacterium tumefaciens bacteria, when it go and infect to the plant cells, then it causes the crown gall disease. You can see here in this figure there is a one crown gall is shown. See this, okay, this is your crown gall. So this causes the crown gall disease. And Agarobacterium rhizogene, it causes the hairy root disease. Hairy root disease means, suppose this is your roots and it causes the hairy root disease, means many hairs comes to this root. And there is another method also, virus mediated method. Yes, Agarobacterium tumefaciens, it is a natural genetic engineer. You have to remember this thing, it is a very important. It is a gram negative soil bacterium. Yes, I told you this. And it naturally transforms the plant cells, and which causes the crown gall. Crown gall, it is what? It is some tumor cell. which is produced to that plant. Okay. Yes. Have you ever gone to the market and bring some coriander to your home, or bring some methi, okay, methi vegetable to your home, then, or spinach vegetable to your home? Then you can observe the root of that plant. Okay, their roots are also come with it, no? So on that root there is a small balls you can see. Okay, so that small balls, what is it? It is a crown gall. Okay, means it is a tumor of that plant, and it is very important. If on that corianders or any plant root, if there is a more number of tumor cells, then there is a more amount of what we can say, minerals or nutrients are present in that plant, or more amount of nitrogen is present in that plant. Okay, so it is very important tip. Yeah. So it infect the plant, this agrobacterium, it infect the plants through the break and the wound. Okay, and is means if suppose that plant is wounded or some break has been there, then this bacteria will infect to that wound or infect to that break. Okay, this tumor formation, it is a result of integration of the tDNA, means transfer DNA, in the plant genome. So this Agrobacterium tumefaciens, it is one type of the soil borne gram negative bacteria. This bacteria, it has its own DNA with it, okay, its own chromosome, and it also have the plasmid. Plasmid means what? Extra chromosomal DNA. It's also have the plasmid, and the name of that plasmid is Ti plasmid. T means tumor, I means inducing, so it is a tumor inducing plasmid. Okay, on that plasmid particular area is present, particular gene is present, and the name of that gene is tDNA. T means transfer DNA, and actually this tDNA, it is transfer from that bacteria to the plant cells. Okay, and when this tDNA is transferred from bacteria to the plant, then that plant will result in the production of crown gall. Okay, means tumor. Yes. Here some points are mentioned which are very important to remember these things, and how this transfer is occur, what is Ti plasmid, we'll see one by one. Okay, this causes the crown gall disease, and Agrobacterium rhizogene causes the hairy root disease. Okay, then curing a particular plasmid and replacing this plasmid with another type of tumorogenic plasmid, it can alter the disease symptoms. Means what? If you take a Agrobacterium bacteria, okay, then you can remove that plasmid, Ti plasmid. After removing, you can insert the another type of the plasmid which also causes the tumor induction, but it does not causes the crown gall, it causes the another type formation of another type of the tumor into that plants. Then classification system, it's divide the genus Agrobacterium into the biovars based on growth and metabolic characteristics. Okay, Agrobacterium can transfer the DNA to remarkably broad group of organism, including the numerous number of the dicots and the monocot angiosperm species. In addition, this can transform fungus also, like the yeast, Ascomyces, Basidiomyces. Okay, and recently this Agrobacterium also reported to transfer the DNA to the human cells. This is a very important thing. Okay, recently it is discovered that Agrobacterium also reported to transfer the DNA to the human cells. There are several vir genes, okay, VIR, virulence genes are present on this Ti plasmid. Okay, from vir A to vir G. Okay, this number of vir genes are present, and we'll see one by one what is the role of that particular vir genes. Okay, yes. Okay, then we'll go for the ti plasmid. Ti means tumor inducing plasmid. Okay, its approximate size is 200 kilo base pairs. Okay, and it is a part of genetic equipment that Agarobacterium tumefaciens and rhizogeny uses to transduce the genetic materials into the plants. The Ti plasmid is lost when Agarobacterium is grown about 28 degrees Celsius. Okay, means if the temperature is very high of particular field or particular soil, then the bacteria which are present in that soil, that is the agarobacterium, they don't have that Ti plasmid. Okay, yes. This Ti plasmid, actually what is present on this Ti plasmid, we'll see with help of the figure, that will be better. Yes, here you can see Ti plasmid is there. It is approximate size is what, 200 kilo base pair, and it has a different types of the genes on it. Okay, so basically we'll see T region. Okay, T DNA or T region, which are responsible for this transfer and all that purpose. So this T region or the T DNA, it is approximately 25 base pair or 25 kb. Okay, yes, it is approximately 25 kb, sorry, 25 base pair. Yes, so what is this tDNA?